Hi, my name is Rondisha Kaiser, and I'm a junior at Oglethorpe University, and I'm currently attending Bio 252. And I want to share before I go into this presentation something exciting about myself. And I will have to think that the most exciting thing about myself is that when I was 16, I had the opportunity to audition for Alvin Ailey Dance Theater. Obviously, I didn't pass the audition. However, I did move to Georgia and graduate high school, and now I am studying biology at Oglethorpe University. Today, my presentation is on serine peptidase inhibitor QNITs type 1. Reduced expression in lemon frost gecko indicates lower survival rate for carcinoma patients. As an overview for this presentation, I will open with an introduction about the background of the research conducted I am presenting on today, the hypothesis of the researchers' research, the experiment methods used to conduct the research, the results, the findings of the researchers regarding the research experiment and hypothesis, and the discussion, where I will apply what the researchers discovered during their experiments to how it impacts the medical field in a meaningful way. Before we go any further into this presentation, I would like to take a moment to explain what is serine peptidase inhibitor QNITs type 1 Spint 1. The serine peptidase inhibitor QNITs type 1 antisense RNA1 Spint 1 AS1 is a long non-coding mRNA and will be referred to as Spint 1 throughout this presentation. In relation to this presentation, Spint 1 is carcinoma and has been identified through homologous mapping of the lemon frost leopard gecko genome. And Spint 1 being identified as carcinoma for the lemon frost leopard geckos is important because the lemon frost leopard geckos were researched by researchers to determine if they will be good models for carcinoma research for humans. And research study named genetics of white color and iridophoma in lemon frost leopard geckos conducted by Longhu and other researchers revealed geckos which are closely related to mammals are a good source for modeling research for carcinoma in humans. Now, the lemon frost leopard geckos are mutant type geckos and they're bred by the following method. A male leopard gecko carrying the lemon frost LF allele, Mr. Frosty, is crossed to 12 female leopard geckos of different genetic backgrounds. In the F1 progeny, which were heterozygous for the LF allele, were back crossed to the same maternal lines or intercrossed to establish a colony of more than 900 animals. Homozygous F2 intercross progeny were named Super Frosties. The hypothesis of the researchers in the genetics of white color and iridophoma in lemon frost leopard geckos is that Spint 1 reduction is associated with negative prognosis in humans with carcinoma, as well as lemon frost geckos are a good model for studying cancer in humans with LF alleles. This hypothesis was supported by the following method: homozygosity mapping, locus of homologous, and transcriptome sequencing. What is linkage mapping? Linkage mapping is a map of genes on chromosomes based on linkage analysis, and it shows relative positions by how often two loci are inherited. It offers evidence that a disease is transmitted from parent to child and linked by one or more genes. In the case of this slide, linkage mapping is being demonstrated between the Mr. Frosty, or also known as the lemon frost gecko, which carries the LF allele, and the sunburst wild-type gecko. The methods used by the researchers to examine the LF alleles within the lemon frost geckos was homozygosity mapping, which variants are closely linked to lemon frost mutations that are expected to have an AF between 0.4 and 0.6 in the lemon frost pool, and in Mr. Frosty, an AF greater than 0.85 in the super lemon frosty pool, and an AF less than 0.15 in the wild type pool. The number of variants meeting this criteria are counted for every 10 kilobytes genome interval. The fraction of such variants among all variants heterozygous in Mr. Frosty within the interval was then calculated. Locus of homologous. Locus of homologous is applications for multi-point linkage analysis are applied to determine the genomic region linked to the diseases. And transcriptome sequencing. Tumor tissues are refractory in RNA extraction, and flanking tumor-free tissue samples are taken from homozygous lemon frost animals, and all samples are homogenized with tissue raptor and buffer RLT immediately after collection. Lysets were immediately frozen on dry ice until the tissue samples were collected from animals, and then all the sets were centrifuged to remove debris, and supernats were taken to fresh tubes, and mRNA was extracted according to the procedures. If you will, put your attention on this slide's illustration, row A. This is a slide illustrating the lemon frost gecko with iridophoma. This slide is also expressing visually how to identify the genetic locus that regulates white color and tumor growth in lemon frost geckos. The researchers used a restriction site associated with DNA sequencing, referred to as RAD sequence, to genotype 188 animals from the breeding pedigree. Now, in box 1, the wild type gecko is represented by the plus plus. The lemon frost gecko is represented by the plus LF allele, and the super lemon frost gecko is represented by a double allele of LF. What this slide is illustrating is that the wild type geckos, although they're positive for tumors, their tumors aren't cancerous. And the lemon frost gecko and the double alleled LF geckos experience metastasis, cancerous tumors. In another study done by researchers titled Decreased Expression of Spint1, AS1, and Spint1 mRNA might be independent unfavorable prognostic indicators in esophageal squamous cell carcinoma. This graph made by Meyer and Kaplan shows the survival rate after surgery and the overall survival rate in comparison to the overall survival rate. And based on Spint1, AS1, and Spint1 mRNA expression levels. The y-axis is the overall survival rate, and the x-axis represents the survival time after surgery. In the study for genetics of white color in iridophoma in lemon frost leopard geckos, 80% of the geckos bred with this phenotype develop iridophoma cancerous tumors by the age of 5 years, and in both male and female gendered reptiles. 900 geckos was utilized in this particular research study, and this is important to know because the researchers hypothesized that the geckos would be good models, serve as good models for research in Spint1. And they also hypothesized that the geckos reduced expression of Spint1. It was related to the poor prognosis of the geckos. In another case study about Spint1, but with human participants, the case study called Decreased Expression of Spint1, AS1, and Spint1 mRNA might be independent unfavorable prognostic indicators in esophageal squamous cell carcinoma. Patients with esophageal squamous cell carcinoma was examined to determine the relationship between Spint1 and prognosis in 99 participants over a 5-year span. With using lemon frost geckos that are bred to carry the LF alleles, scientists are able to further investigate with more models of lemon frost geckos than in humans. And unfortunately, of the 99 humans over a 5-year span, research can't keep up. The research isn't moving fast enough to come up with something to disrupt Spint1 expression to slow the cases of cancer growth or eliminate the cases of cancer growth using human participants. However, using a gecko model to serve as a model for how the cancer grows, and if the growth of cancer can be eliminated or disrupted, is cost effective. It's not only cost effective, it provides an abundance of models to research. So, researchers conclude lemon frost geckos are good models for carcinoma research in humans, mammals, and other reptiles because of Spint1's prevalence in the mutant breed. Researchers determined Spint1 reduced to be associated with poor prognosis of carcinoma in patients. My name is Rondisha Kaiser, and I just presented for the reduced expression of Spint 1, it being a poor prognosis for carcinoma. I hope that this presentation was understandable. I hope that you recognize the significance of using lemon frost geckos, mutant types, to continue research for carcinoma patients. Um, and realize that there is a significant amount of mutant type geckos available opposed to utilizing human participants in carcinoma research. Thank you and have a good day.